Good morning, students. We will start with metabolism of carbohydrate. Today's competency is competency number 3.2 and today's topic is digestion and absorption of carbohydrates. Dietary carbohydrates means the carbohydrates present in our diet are in the form of polysaccharide, disaccharide and monosaccharides. Polysaccharides found in our diet is mainly starch, which forms bulk of our diet. Disaccharides present in the diet are sucrose, which is a table sugar or common sugar, and lactose, which is also called as milk sugar. Monosaccharides are also found in very small quantity in our diet, and these are glucose, fructose and galactose. Disaccharide and polysaccharide require digestion, whereas monosaccharides does not require digestion, they are directly absorbed into the blood. Digestion of carbohydrate takes place in the gastrointestinal tract with the help of various enzymes. First we will see digestion of carbohydrates. Digestion of carbohydrates starts in mouth. In the mouth, the enzyme salivary amylase is found in saliva, which act on starch and break the starch into maltodextrins and dextrins. pH required for the activity of salivary amylase is neutral, whereas it also requires chloride ions for its activity. The digestion of starch by salivary amylase in mouth is incomplete because time available for the digestion of salivary amylase in mouth is very less. Hence, incomplete digestion of starch takes place with the help of salivary amylase. This is the diagrammatic representation of structure of starch. In this figure, every circle indicates a glucose molecule. These glucose molecules are joined together by two types of linkages. One is alpha 1,4 glycosidic linkage and another is alpha 1,6 glycosidic linkage. When two glucose molecules are joined together by alpha 1,4 glycosidic linkage, they form straight chain structure. But when a branching point is formed, or when the branch originate from the existing branch, that branching point glucose molecule is joined by alpha 1,6 glycosidic linkage. So in this figure, the dark circle indicates alpha 1,6 glycosidic linkage. Next come to digestion in stomach. Practically no digestion takes place in the stomach. Next, digestion in small intestine. In the small intestine, two types of juice are present which are responsible for digestion of carbohydrates. These juice are pancreatic juice and intestinal juice. These juice contains various enzymes. First we will see pancreatic juice or digestion by pancreatic juice. Pancreatic juice contains enzyme called as pancreatic amylase. This pancreatic amylase differs from salivary amylase structurally, but both have same function. Therefore, they are isoenzymes of each other. At the same time, salivary amylase and pancreatic amylase are synthesized by two different genes. Therefore, salivary amylase and pancreatic amylase are called as true isoenzymes as they are the product of two different genes. Now come to the action of pancreatic amylase. Pancreatic amylase also acts on starch, breaks the alpha 1,4 glycosidic linkage and convert the starch into maltose, maltotriose and dextrins. Here dextrin is nothing but it has similar structure to the starch, but branches are short as compared to starch. Now this pancreatic amylase requires pH 7.1 for its activity. It also requires chloride ions. Pancreatic amylase breaks all starch molecule into product. Therefore, this digestion is called as complete digestion of starch. Now the second juice is intestinal juice, which contains the following enzymes: maltase, trihalase, dextrinase, sucrase and lactase. Maltase acts on maltose, breaks the alpha-1,4 glycosidic linkage and convert it into glucose molecule. Trihalase acts on maltotriose and convert it into glucose molecule, whereas dextrinase acts on dextrin, which breaks both alpha-1,4 as well as alpha-1,6 glycosidic linkage and form product like glucose, maltose and maltotriose. So in this way, the starch is converted into glucose by the action of various enzymes that we have studied. Now the next enzyme present in intestinal juice is sucrase, which acts on a disaccharide sucrose and break it into glucose and fructose. Another enzyme lactase acts on lactose and convert that lactose into glucose and galactose. So product of digestion of carbohydrates are glucose, main product, fructose and galactose. So in this way, polysaccharide, that is starch, and the disaccharide sucrose and lactose present in the diet are converted into monosaccharide. So this completes the digestion of carbohydrate in small intestine. Now come to absorption of carbohydrates or absorption of monosaccharides properly. Absorption of monosaccharides takes place in two phases. In the first phase, monosaccharides are absorbed from gastrointestinal lumen into epithelial cell, and in the second phase, they are absorbed from epithelial cell into the capillary. This figure shows the diagrammatic representation of absorption of monosaccharides. First GIT lumen to epithelial cell and from epithelial cell to blood. Glucose, fructose and galactose are transported from the lumen of GIT to the epithelial cell with the help of two types of transporters. First is sodium dependent glucose transporter, short form is S-GLUT, and second is glucose transporter 5 or GLUT5. So in this figure you can see S-GLUT and GLUT5. First we will see the action of S-GLUT or sodium dependent glucose transporter. Now this sodium dependent glucose transporter has two different sites. One for binding of sodium and another site is for binding of glucose or galactose. So one monosaccharide is bind at one side and sodium ion is bind to the another side. When these two binds to the S-GLUT, there is a conformational change in the movement of S-GLUT takes place and it rotates to 180 degree and opens inside the epithelial cell. When it opens into the epithelial cell, monosaccharide, either glucose or galactose, which is attached there, will be released. At the same time, sodium ion is also released into the epithelial cell. Here you have to note that this S-GLUT is transporting a glucose molecule against the concentration gradient, because the concentration of glucose inside the epithelial cell is more as compared to lumen of GIT, but it transports the sodium ion along the concentration gradient because the concentration of sodium ions inside this epithelial cell is less as compared to lumen. Sodium, all of you know, sodium is a extracellular ion. It is not intracellular ion. So concentration of sodium inside the cell is less. So S-glut is transporting glucose and sodium ion at a time. So it is also called as co-transport. The important thing you have to note is glucose, it is transporting glucose against the concentration gradient and sodium along the concentration gradient. But here it does not require energy for this transport. When the glucose or galactose is released into the epithelial cell, sodium is also released inside the epithelial cell, but sodium is an extracellular ion. It is pumped outside the epithelial cell by sodium pump on other side of the epithelial cell. To pump this sodium ion from the epithelial cell to outside the epithelial cell, this sodium pump utilizes 1 ATP. So 1 ATP molecule is used at this stage. If you summarize the action of S-glut, then epithelial cell has got 1 glucose molecule and it has utilized 1 ATP molecule. Sodium is pumped outside. So what is gain for epithelial cell? Gain for epithelial cell is 1 glucose molecule and it has lost 1 ATP molecule. So transport of glucose or galactose by sodium dependent glucose transporter is indirectly active. Therefore, such type of transport is also called as secondary active transport. Now come to GLUT5. GLUT5 transports mainly fructose. It is poor transporter of glucose. It's bind with the fructose on one side, and when the fructose binds, it undergoes conformational change, and there is rotation of this GLUT5 protein to 180 degree, and it opens inside the epithelial cell, and the fructose is released inside the epithelial cell. This transport does not require energy, and it transports only 1 molecule at a time. So it is called as uniport type of transport, and as it does not require energy, so it is called as facilitated diffusion. So with the help of these two types of transporter, glucose, galactose and fructose have entered into the epithelial cell. Now the next step is the transport of these monosaccharides from epithelial cell to the capillaries, which are present on other side of the epithelial cell. These 3 monosaccharides are transferred to the blood compartment by only one type of transporter, which is called as glucose transporter 2 or GLUT2. Now this GLUT2 binds with the glucose, galactose and fructose one by one, and it rotates to 180 degree so that it can open on outside the epithelial cells. So this is also a uniport type of transport because it will transport 1 molecule at a time, and as it does not require energy, so it is a facilitated diffusion. This is the summary of various glucose transporters present in various tissues of our body and their properties. We have studied glucose transporter 2, we have studied glucose transporter 5, and we have studied sodium dependent glucose transporters. So this slide shows the same thing that I have already explained. Sodium dependent glucose transporter is also called as secondary active transport. Why it is called as secondary active transport, I have explained. It transports glucose or galactose along with sodium and the sodium is later on expelled by sodium pump by using energy. Glucose transporter transport the glucose and galactose against the concentration gradient, but sodium along the concentration gradient. Same thing, glucose transporter 5 will transport the fructose from lumen of epithelial cell to the inside the epithelial cell. It is called as facilitated diffusion. Same, epithelial cell, glucose transporter 2 present on another side of the epithelial cell will transport the monosaccharides from epithelial cell to outside the epithelial cell. Transport by glucose transporter 2 is not sodium dependent, it is unipod, facilitated type of diffusion. So this is figure shows the facilitated type of transport. Here see that monosaccharide binds to the transporter. First the transporter opens on one side. When the monosaccharide binds, then they get close that side and opens on another side. So in this way the monosaccharides are transported by facilitated transporter like glucose transporter 2 and glucose transporter 5. So in this way the monosaccharides are absorbed from GIT. Now come to clinical application. In oral rehydration fluid, ORS, oral rehydration solution, consists of glucose and sodium, which is used in case of treatment of diarrhea. Why glucose is added to this ORS solution is, all of you know, sodium dependent glucose transporter will transport sodium and glucose. It will not transport sodium alone. Therefore, we have to add glucose into the oral rehydration solution so that sodium is easily reabsorbed from GIT. This completes the digestion and absorption of carbohydrates. Thank you.